Welcome to the Deep Dive, where we cut through the noise and get straight to the fascinating core of complex topics. Today, we're diving into a concept that sounds, well, straight out of a sci-fi thriller: zombie cells. Imagine cells in your body that refuse to die, just lingering around and quietly wreaking havoc as you age. Now, what if we could actually clear them out? That's our intriguing starting point for this deep dive. We've got a stack of really cutting-edge research in front of us. We're exploring a revolutionary science called senolytics, and it's surprising, pretty profound impact, especially on our beloved pets.
Right. And our mission today is, well, to extract the most important insights from all this material. We'll unpack how these therapies are fundamentally reshaping our understanding of aging, particularly within the brain, and, how they promise to extend the healthy lives of our companion animals. What's truly fascinating here, I think, is how this fundamental cell biology is translating directly into practical applications for vet medicine. It's actually leading the way in some aspects of this research.
Absolutely.
Yeah.
So whether you're just curious about the future of anti-aging or, you're a devoted pet parent looking for those aha moments and maybe some practical takeaways for your furry family members' health, you were definitely in the perfect place. Let's dig in. Okay, so let's start with the basics. What exactly are these zombie cells we keep hearing about?
Okay, so the scientific term is cellular senescence. Think of it as an irreversible state where a cell stops dividing. But here's the kicker: it doesn't die. It just hangs around, metabolically active, like a persistent squatter in your tissues.
Oh.
We call them zombie cells because they're essentially stuck, between life and death. They're not doing their normal jobs, and worse, they're actively damaging their surroundings.
And how do they happen? Is it just part of getting older?
Well, yes, it's definitely associated with aging. Cells can enter the senescent state in a couple of main ways. One is called replicative senescence. That happens as telomeres, those little protective caps on the ends of our chromosomes, they get shorter with each cell division.
Right.
Eventually, they get critically short, and the cell just stops dividing. The other major trigger is stress-induced premature senescence. So things like DNA damage, oxidative stress, even activating certain cancer genes that can prematurely trigger this state.
Yeah.
It's basically the cell's way of hitting the brakes when something's gone seriously wrong.
That makes sense. So if a cell is damaged, it stops dividing.
Yeah.
Smart. But why do these zombies build up more as we age?
Yeah, what's fascinating here is how our body's cleanup crew responds. See, in younger, healthier bodies, the immune system is usually pretty efficient at clearing these senescent cells away.
Okay.
But as we age, that cleanup crew, our immune system, it starts to decline. We call it immunosenescence.
Immunosenescence, right?
Yeah. So you get this double whammy: an increasing rate of senescent cell formation from accumulated damage, plus a reduced ability to actually remove them. This leads to them building up, accumulating in basically all tissues, and that drives functional decline in organs. And if you connect this to the bigger picture, this accumulation, it's now understood to be a fundamental, actually a causal driver of aging itself.
Wow.
It's not just a marker of getting old, it's actively making it happen.
Okay, this is where the true menace of these zombie cells really comes into focus. It's not just that they're hanging around doing nothing. It's what they do that's so destructive, right? They get this thing called the senescence-associated secretory phenotype, or XASP. Tell us about that.
Exactly. SASP is essentially their toxic secret weapon. It's this relentless chemical barrage, a toxic cocktail of pro-inflammatory molecules.
Okay.
These are tiny chemical messengers, things like interleukins 6 and 8, chemokines that call in immune cells, and proteases, enzymes that actively break down the healthy tissue around them. They just pump this stuff out constantly.
So they're not just inert, they're actively poisoning the well for the healthy cells nearby.
Precisely. And the harmful effects of this SIP are, well, profound and far-reaching. This cocktail causes chronic low-grade inflammation throughout the body, that state often called inflammaging.
Inflammaging, heard that term.
Yeah. These secreted molecules can directly damage healthy surrounding cells, messing up their function. They also have those proteases that degrade the tissue matrix. Think about cartilage deteriorating in osteoarthritis, or skin losing elasticity, getting wrinkles. And even more insidiously, these SASP factors can actually spread senescence to healthy neighboring cells.
No way.
Yeah, creating this vicious cycle that amplifies and spreads the damage. And critically, this pro-inflammatory environment created by the SASP, it's directly implicated in a huge range of age-related diseases: cardiovascular problems, fibrosis in organs, and really key for our discussion today, neurodegenerative diseases like Alzheimer's.
Wow, okay.
Right.
Given this destructive cascade, the obvious question becomes, can we actually get rid of these zombie cells? Is there a way?
That's exactly where senolysis comes in. It's the selective pharmacological elimination of these senescent cells. The aim is to stop or maybe even reverse these pathological processes.
Okay.
And this represents a real paradigm shift in medicine, you know? Instead of just treating individual age-related diseases one by one, senolytics offer the possibility of targeting a common root cause. This approach aims to modulate the fundamental mechanisms of aging itself, promoting what we call health span.
Health span. So not just living longer, but living healthier longer.
Exactly. The period of life spent free from chronic disease. That's the goal.
That sounds incredibly promising. But wait, does senescence always have a negative role? Didn't you say it helps stop tumors sometimes or helps with wound healing?
That's an excellent point, and it raises a really important question for senolytics. Senescence does have crucial beneficial roles, especially in acute situations, like stopping a damaged cell from becoming cancerous or sealing off a wound. You want that quick stop then. The answer seems to lie in the concept of intermittent dosing. The goal isn't to wipe out all senescent cells all the time. It's to periodically clean out the accumulated chronic ones, the ones that are persistently causing trouble over the long term, without interfering with the body's ability to use acute senescence when it's needed for repair or tumor suppression.
Ah, okay. Like a periodic spring clean, not a continuous demolition.
Exactly. That's a good way to put it.
So let's talk about the specific compounds, the actual ghostbusters for these zombie cells, the senolytics themselves. What are we looking at?
Well, the first major breakthrough in this field came with the discovery that a combination of two existing compounds, known as D plus Q, could effectively eliminate senescent cells.
D plus Q.
Yeah, that's dasatinib, which is actually a chemotherapy drug combined with quercetin, which is a natural flavonoid found in many plants. What was really crucial about this initial finding was that these two agents work on different types of senescent cells.
Oh, interesting.
It highlighted that zombie cells aren't all the same. They're heterogeneous. This showed that different senescent cells might rely on different survival tricks, meaning we might need drug cocktails like D plus Q for broad-spectrum clearing.
Okay, so dasatinib is a chemo drug. Are we mostly talking about powerful pharmaceutical drugs here, or are there more natural options, especially thinking about pets?
Absolutely. While synthetic drugs like dasatinib are potent, their potential toxicity for long-term anti-aging use has definitely driven intense interest in natural senolytics. Compounds with more favorable safety profiles, which is, like you said, particularly relevant for you, our listener, and your pets.
Right.
Ficitin, for example, it's a powerful flavonoid found in high concentrations in strawberries, apples, mangoes. It's been identified as one of the most potent natural senolytics in preclinical studies.
Ficitin. Okay.
Then there's quercetin, which we just mentioned in the D plus Q combo. It also has senolytic effects on its own, and it's abundant in things like onions, broccoli, capers. Curcumin, the active compound in turmeric, has also shown senolytic activity, along with its well-known anti-inflammatory property.
No way.
Yeah. And there are others being explored too. Apigenin from parsley and chamomile, EGCG from green tea, silymarin from milk thistle. Quite a few natural compounds.
It sounds like nature has some pretty powerful tools in its arsenal here. What about how they work? How do these compounds actually tell a zombie cell to, finally die?
That's a great question. The core mechanism is that senolytics target the acquired vulnerabilities of senescent cells. See, these zombie cells, to stay alive when they shouldn't, actually develop this whole network of pro-survival pathways to resist programmed cell death or apoptosis.
Apoptosis. Cell suicide.
Exactly. Senolytics act by selectively deactivating key parts of these defense networks. They essentially cut off their life support, and that then pushes the senescent cells into apoptosis.
So they exploit a weakness.
Sort of. For example, some senolytics inhibit the BCL2 family of anti-apoptotic proteins. Blocking those allows the pro-death signals to take over. Dasatinib, on the other hand, works differently. It disrupts the internal communication system that zombie cells rely on to stay alive, blocking the signals that keep them from dying.
Okay.
This diversity in how they work shows there's no single death switch. Different senolytics target different vulnerabilities these cells have developed.
So we have the theory, the why, and we have the tools, the how. What's the real-world evidence? Does this actually work in living organisms?
Yeah, the promise of senolysis is definitely rooted in some pretty solid preclinical evidence, mostly from mouse models so far. Pioneering studies where they genetically engineered mice so they could eliminate senescent cells on command showed conclusively that this delayed or lessened a whole range of age-related problems, things like kidney disease, heart problems, even cancer. Then, pharmacological studies confirmed these findings. There was a landmark study from the Mayo Clinic. They showed that intermittent treatment of older mice with that D plus Q combination significantly improved physical function, reduced frailty, and remarkably, extended their median lifespan by 36%.
36%, that's huge.
It is, and crucially, they didn't just live longer, they lived healthier. They compressed that period of sickness at the end of life.
That health span idea again.
Exactly. And another striking finding, they found that even transplanting a relatively small number of senescent cells into young, healthy mice was enough to cause physical problems and reduce their survival.
Just transplanting them.
Yeah, it powerfully demonstrated just how potent their negative impact is.
Okay, and while human trials are underway now for various conditions, you hear about them for lung fibrosis, diabetes, osteoarthritis, it's still very much experimental for human use, right?
Absolutely. Early days for humans.
Which is where the connection to our pets becomes even more compelling and really hits home for you, the listener, and your furry family members. Why are dogs, specifically, such an invaluable model for aging research?
Dogs are just exceptionally powerful in translational research models. It's quite amazing, really.
Yeah.
Unlike lab animals kept in sterile cages, dogs share our home environment, our exposures to pollutants, often our lifestyles, our diets.
Puppy live with us.
They also have considerable genetic diversity, much like humans, which means findings are more likely to be broadly applicable. And most importantly, they naturally develop many of the exact same age-related diseases that humans do.
Like what?
Osteoarthritis, heart and kidney disease, various cancers, and crucially for this discussion, a form of senile dementia, CCDS, that closely mirrors human Alzheimer's disease.
CCDS, okay.
And because their lifespan is shorter, the whole aging process happens on an accelerated timeline. So longitudinal studies on anti-aging interventions that would take decades in humans, you can complete them in just a few years in dogs. It massively accelerates discoveries that ultimately benefit both animals and humans.
That makes so much sense. So given they age like us, what senolytics are being explored specifically for pets right now? You mentioned those natural compounds.
Yeah, you're right to focus on the natural compounds like fisetin and quercetin. Their natural origin and established safety profiles in vet use make them really attractive.
Okay.
Quercetin, for instance, is already pretty well known as a nutraceutical in veterinary medicine. Vets use it for its anti-inflammatory, antioxidant, and histamine properties, often as a supplement for things like allergies or joint pain.
Right, I've seen that.
And while its current marketing doesn't always shout about the senolytic action, its proven safety and multiple benefits make it an ideal candidate. Fisetin, though, is rapidly emerging as a major player specifically for its senolytic action. There are already veterinary supplements on the market formulated with fisetin, and they're marketed directly for that purpose to help address geriatric issues. Things like improving neurological function, maybe reducing cancer risk, mitigating chronic diseases in older dogs.
That's great to hear these are actually available or becoming available. So if a pet owner is thinking about this, looking for these supplements, what should they be aware of? Any cautions?
That's a really vital question. While these compounds are promising, it is absolutely crucial for pet owners to talk to their veterinarian before starting any new supplement.
Definitely.
Dosage can vary a lot based on the pet's size, age, existing health conditions. A vet can guide you to reputable brands. They can discuss potential interactions with other medications the pet might be on and help monitor for any side effects.
Good advice.
It's all about making informed choices, always with veterinary guidance. And what's truly groundbreaking here is the scale of the clinical trials happening in dogs right now. It really rivals human studies in some ways. For example, there's the Triad Dog Aging Project funded by the NIH. They're investigating rapamycin.
Rapamycin, yeah, I've heard of that one.
It's a compound that helps regulate cellular growth, and they're looking at it for extending healthy lifespan in dogs. Huge study. Then there's the STAY study by a company called Loyal. That's a massive trial, over 1,300 dogs, I think. They're aiming for FDA conditional approval for a drug for healthy lifespan extension by 2026.
Wow, FDA approval for lifespan extension, that's huge.
It's a monumental regulatory precedent. It really shows how veterinary medicine is pushing the boundaries here.
Amazing.
And finally, the Leap Years LYD62 trial, which we'll definitely dig into more, that's specifically focused on cognitive function. It used a combination of a senolytic and an NAD plus precursor that's a compound that helps boost cellular energy levels, which decline with age.
Okay, so multiple big trials looking at different angles. It really suggests the future of healthy aging for pets, and maybe for us too, is probably going to involve a multi-pronged approach, right?
Absolutely. Combining several strategies to tackle the different facets of aging seems like the most promising path forward.
Okay, let's zoom in on one of the most heartbreaking parts of aging in pets, something many listeners might sadly be familiar with: cognitive decline. Let's deep dive into canine cognitive dysfunction syndrome, or CCDS. You mentioned it's often called canine Alzheimer's.
Yeah, CCDS. It's far more than just a dog getting a bit old and forgetful. It's a progressive neurodegenerative disorder, and the parallels with human Alzheimer's disease are really quite striking, neuropathologically speaking.
Oh, so.
Well, at a microscopic level, if you look at the brains of dogs with CCDS, you see the same characteristic hallmarks: deposits of beta amyloid protein plaques and tangles of tau protein.
The same plaques and tangles as in human Alzheimer's.
Exactly the same. And as the disease progresses, you also see macroscopic changes. The brain cortex shrinks, the ventricles enlarge, there's a general loss of neurons. And crucially, recent research has really zeroed in on senescent cells, particularly glial cells. Those are the support cells in the brain, like astrocytes and microglia accumulating in the aging brain.
The zombie cells are in the brain too.
They are. And these senescent glial cells adopt that nasty SASP we talked about earlier. They release this flood of pro-inflammatory cytokines right into the brain's environment.
Okay.
This creates chronic neuroinflammation, which is a central feature of both CCDS and human Alzheimer's. This inflammation damages neurons, messes up communication between them, actually promotes more amyloid and tau deposition. And it can even spread senescence to neighboring glial cells, just amplifying the whole pathological process.
Wow, it really feeds itself.
It does. And beyond inflammation, the aging brain also struggles with metabolic dysfunction specifically, a significant drop in glucose metabolism. The brain isn't getting enough energy, and that compromises cognitive function too.
So what signs might an owner actually see if their dog is developing CCDS? How can they spot it?
Yeah, there's a helpful acronym vets often use, DISHA, or sometimes DISHA,
Yeah.
that covers the common behavioral changes owners might notice.
Disha.
D is for disorientation. Your dog might seem lost in familiar places, maybe stare blankly at walls or get stuck in corners.
Okay.
S is for interaction changes. They might show altered social behavior, less greeting, maybe less interaction with family members or other pets. S is for sleep-wake cycle disturbances. It's really common for them to sleep more during the day, but then be restless, wander around, or even vocalize at night.
Oh, the nighttime wandering, yeah.
H is for health habits or learning. They might forget previously learned commands, or sadly, start having accidents inside the house again after being perfectly trained for years.
That must be tough.
A is for activity changes. This can be a general decrease in activity, less interest in playing, or sometimes, weirdly, a purposeless increase in activity, like pacing or circling.
Pacing, right?
And often there's an additional A for anxiety. You might see increased generalized anxiety, new phobias developing, or just more irritability.
Okay, Disha. That's a useful checklist.
It is, and the veterinarian's role here is absolutely crucial for diagnosis. First step is always ruling out other medical conditions that could cause similar signs, things like chronic pain from arthritis, vision or hearing loss, thyroid problems, other diseases.
Right. Rule out other causes.
Exactly. Vets use standardized questionnaires like the Canine Cognitive Dysfunction Rating Scale, the CCDR scale, to help owners score the frequency and severity of these signs. Traditionally, management has been mostly palliative supportive care, environmental enrichment, maybe dietary changes with antioxidants, some medications that might slow things down a bit, but no real cure.
Until perhaps now. Let's talk about that game changer. The 2024 LYD62 clinical trial. This sounds like where the research takes a really profound turn for pets.
It really does. This trial represents the first rigorous, controlled evidence that a therapy directly targeting senescence can effectively treat dementia in a higher mammal, in dogs.
Okay, tell us about it.
So the study recruited 70 senior dogs, all 10 years or older, who had confirmed mild to moderate cognitive decline based on that CCDR scale. It was designed as a randomized, double-blind, placebo-controlled trial that's the gold standard for clinical research, right?
Yeah.
No one knew who was getting what.
Right, gold standard.
The intervention, called LYD62, was a combination therapy. It involved an intermittent senolytic derived from plant flavonoids like fisetin and quercetin, remember those, given just 2 consecutive days per month.
Only 2 days a month.
Yeah, that intermittent spring clean idea. And that was combined with a daily NAD plus precursor supplement to help boost cellular energy. So a dual approach targeting both the senescent cell burden and the age-related decline in cellular energy metabolism.
Okay, makes sense. And the results? Did it actually work? What did they find?
The key finding was a statistically significant improvement in the owner-assessed CCDR scores. A really remarkable 88.9% of the dogs treated with LYD62 showed an improvement in their cognitive function, compared to 60% in the placebo group.
Wow, nearly 90% improved versus 60%. That's a big difference.
It is. And importantly, the treatment was very well tolerated. There were no significant differences in adverse events between the groups. This trial, it fundamentally shifts our understanding. It suggests canine dementia isn't just an inevitable part of aging anymore. It might actually be a treatable condition, opening the door to therapies that truly modify the disease process.
That's absolutely monumental.
Yeah.
But do they see similar improvements in, like, objective cognitive tests done at the clinic? Or was it mainly based on what the owners were seeing at home?
That's a really crucial question, and it points to an ongoing challenge in this field. While that improvement in owner-assessed CCDR scores is paramount, that reflects the dog's quality of life at home, which is what matters most to owners.
True.
The objective cognitive test performed in the clinic didn't show the same statistically significant difference between the groups. It's also worth noting that all the groups, including the placebo group, showed some level of improvement.
The placebo effect.
Yeah, suggesting either a strong placebo effect or maybe just the benefit of the increased attention and care pets get when they're enrolled in a clinical trial.
Right.
This highlights a persistent challenge in veterinary behavioral medicine. How do you objectively measure cognition reliably in nonverbal patients? While owner perception of quality of life is ultimately key, future research definitely needs to develop more sensitive, objective assessment tools that can corroborate owner reports and give us a more complete picture of treatment efficacy.
Got it. So promising results on the owner side, but more work needed on the objective measures. Still a huge step forward. The success of that LYD62 trial in canine cognition is remarkable. But this deep dive, it seems to reveal an even broader picture. It suggests brain health isn't just about what's happening inside the skull, right? There's this vital, often overlooked connection emerging, the gut-brain axis.
Absolutely. The gut-brain axis is this incredibly complex, bidirectional communication system. It connects the emotional and cognitive centers of the brain directly with the peripheral functions of the gut, and importantly, the trillions of microbes living there, the gut microbiome.
Bidirectional, so it goes both ways.
Exactly. Communication flows constantly in both directions. It happens through multiple pathways. There are neural connections, like the vagus nerve, which runs directly from the gut to the brain. There are immune pathways involving cytokines and immune cells, and endocrine pathways, where gut bacteria actually produce neurotransmitters like serotonin and GABA or other metabolites that can travel through the blood and influence brain function. It's a continuous conversation.
Wow. So how does an imbalanced microbiome, what you call dysbiosis, how does that affect our dog's behavior and cognition then?
Well, an imbalanced gut microbiome, dysbiosis, has been increasingly linked to a whole range of health problems. And that includes behavioral issues like anxiety and aggression, and definitely cognitive decline in aging dogs.
How does that work?
Dysbiosis can lead to something called leaky gut, or increased intestinal permeability. This means the gut barrier isn't as strong, allowing bacterial components and inflammatory molecules to leak out of the gut and into the bloodstream.
Okay.
This triggers systemic, body-wide, low-grade inflammation. And that inflammation can then cross the blood-brain barrier and exacerbate neuroinflammation in the brain. It can also alter neurotransmitter production and function, contributing directly to things like anxiety and cognitive problems.
So leaky gut leads to leaky brain, essentially.
That's a good way to think about it, yeah. The connection to the aging brain is particularly strong. Studies in animal models have shown that altering the microbiome contributes to Alzheimer's-like pathology. And remarkably, doing fecal microbiota transplants, basically transferring gut microbes from young, healthy donors to older recipients, has actually shown promise in improving cognitive function in some animal studies.
Fecal transplants for brain health. That's wild. It really highlights the connection.
It really does.
Okay, so this leads to a fascinating therapeutic intersection. Can we actually target the gut to improve brain health in our pets?
Absolutely. Recognizing the power of the gut-brain axis has opened up totally new therapeutic avenues. Manipulating the microbiome through diet, Prebiotics, which feed good bacteria, and probiotics, which introduce good bacteria, is becoming a much more standard strategy now.
Right, probiotics are everywhere.
Yeah, and certain probiotic strains, like Bifidobacterium longum BL999, have even been shown to act as psychobiotics.
Psychobiotics.
Yeah, probiotics that have a specific beneficial effect on mental health. That particular strain has been shown to help dogs maintain calm behavior and reduce anxiety. They work by restoring microbial balance, strengthening that gut barrier, and even producing neuroactive compounds themselves.
Okay, that makes sense. But what's truly fascinating here, you're saying, is the potential link between the senolytics we've been talking about and gut health.
Exactly. This is a really exciting emerging area. Recent research indicates that some of those senolytic flavonoids we discussed, quercetin, quercetin, curcumin, can directly modulate the gut microbiome composition in a beneficial way.
Wow.
For example, quercetin has been shown in studies to increase the abundance of beneficial bacteria like Bacteroides, Bifidobacterium, and Lactobacillus in the gut. And at the same time, it helps reduce intestinal inflammation and strengthen that gut barrier. Quercetin and curcumin seem to have similar positive effects.
Whoa! So this suggests a potential double-hit mechanism for these senolytics then. They're directly clearing senescent cells in the brain, reducing neuroinflammation there. And they might be indirectly improving brain health by fostering a healthier gut microbiome and reducing systemic inflammation coming from the gut.
That's a very compelling hypothesis, yes. This potential dual action hitting both the brain directly and the gut-brain axis might help explain the remarkable efficacy seen in that LYD62 trial for CCDS. It might not have just been about clearing zombie cells in the brain. It might also have been about restoring gut-brain axis health at the same time.
That's fascinating.
It really opens up an exciting new avenue for research, maybe exploring combinations of senolytics with specific psychobiotics to see if we can get even better results. It just highlights how interconnected everything is.
Truly.
Yeah.
Okay, as we wrap up this deep dive, let's look towards the horizon. This new era of longevity medicine, it brings immense promise, but there must be challenges too, right?
Oh, absolutely. There are several significant obstacles to widespread clinical adoption, for sure. First, there's the regulatory pathway. It's basically uncharted territory. There's no clear precedent for approving drugs specifically for healthy lifespan extension, especially in beat med, although things are moving.
Right, that FDA approval Loyal is seeking would be huge.
Exactly. Companies like Loyal are actively working with the FDA's Center for Veterinary Medicine to figure out what safety and efficacy data is needed. Their success in gaining that preliminary reasonable expectation of effectiveness designation is a crucial step, but final approval still requires robust, long-term data, which takes time.
Okay, regulatory hurdles. What else?
Well, scientifically, we still face challenges because, as we said, senescent cells are heterogeneous. They're not all the same. Their markers, their survival pathways, they can vary depending on the cell type and the tissue. And their role can be context-dependent, sometimes helpful, sometimes harmful.
Right.
This means we likely need more selective senolytics, drugs that target only the bad chronic senescent cells, and we need to optimize dosing strategies carefully. And then there are the practical and economic challenges.
Cost, I imagine.
Cost is definitely one. The long-term cost of these new drugs could be a barrier for many pet owners. And also, objectively measuring improvements in quality of life in individual patients. It's inherently complex, especially in animals who can't tell us how they feel. We often rely heavily on subjective owner perception, which is valuable, but we really need to develop and adopt more standardized, objective assessment tools.
Makes sense. So what else is on the horizon? Beyond just senolytics, what other approaches are part of this burgeoning field of geroscience?
Yeah, senolytics are definitely one of the most exciting strategies right now, but they're part of a much broader field. The future of veterinary longevity medicine will likely involve a multimodal approach, combining several different strategies.
Like what?
Well, things like stem cell therapies. They're already used in vet med, mainly for osteoarthritis, for tissue repair and inflammation modulation. That can expand. Gene therapy is also being explored. For example, there are trials looking at SIRT6 gene therapy in dogs. SIRT6 is part of a family of proteins involved in longevity and DNA repair.
Gene therapy for aging. Wow.
Yeah. And then there are metabolic modulators. We mentioned rapamycin being studied in the dog aging project that affects the mTOR pathway involved in cell growth and metabolism. And another drug called acarbose, which affects blood glucose levels, has shown promise in extending lifespan in lab animals and is also being investigated in dogs.
So a whole toolkit is developing.
Exactly. It's likely going to be about combining approaches tailored to the individual.
Okay, this ability, or potential ability, to significantly extend a pet's life. It brings up some pretty profound ethical considerations, doesn't it? It's not just about what we can do, but what we should do for our companions.
That's an absolutely crucial point, and it's something veterinarians and pet owners will increasingly need to grapple with. The core ethical imperative here really has to be the distinction between health span and mere lifespan.
Right. We keep coming back to that.
The primary goal of longevity medicine should never be simply to add years to life, regardless of the quality of those years. It has to be about adding life to years. The moral justification for these interventions really lies in their potential to compress morbidity, to shorten that period of illness and suffering at the end of life.
So preventing suffering is key.
Absolutely. Prolonging a life filled with chronic pain, severe dementia, or incapacitating frailty. That would be ethically problematic. It would go against a veterinarian's fundamental duty to alleviate suffering.
So it really comes down to making sure those extra years are good years, quality years.
Exactly. And that necessitates rigorous, continuous assessment of the animal's quality of life, which, as we've said, is challenging in nonverbal patients. It requires vets and owners to work together to carefully interpret behavioral and physical signs to really determine if the animal's life is still enjoyable, still meaningful from the animal's perspective.
That sounds difficult.
It is. The veterinarian has to act as the patient's advocate, sometimes guiding owners to prioritize the animal's well-being over, perhaps, a human desire to postpone grief.
Yeah.
It really calls for strong ethical training and very advanced communication skills within the veterinary profession. It's a balance we're all still learning to strike as these powerful new capabilities emerge.
What a deep dive this has been into the really fascinating world of senolytics and pet longevity. We see how these mysterious zombie cells seem to drive the aging process, how targeted therapies are being developed to remove them, and the truly groundbreaking impact this is already starting to have on our furry friends, especially in tackling heartbreaking conditions like canine dementia, and maybe even influencing their gut health too.
Yeah, knowledge is most valuable when it's understood and, applied thoughtfully. This field is moving incredibly fast. And what's happening right now in veterinary medicine, as we've seen, is directly informing human geroscience and vice versa. It really underlines that there's always more to learn and that critical thinking is just essential as we navigate all this exciting new information.
Absolutely. So your mission, should you choose to accept it, is maybe to consider this. As we gain this incredible power to potentially extend life, for both ourselves and for those non-human companions we love, how do we truly define a good life? And how does that understanding shape the choices we make for those we hold most dear? Keep that thought with you until our next deep dive.